let me just, yeah, thank you. So I'll just repeat what I just. So we are pleased to have Dr. Amit Dutt as our next speaker. Dr. Amit Dutt is a basic scientist at ACTREC at Tata Memorial Center, Mumbai. Dr. Dutt obtained his PhD from the University of Zurich, Switzerland, and ICGEB/JMI, New Delhi. He was a postdoctoral fellow at the Broad Institute of Harvard and MIT, and his lab focuses on understanding somatic changes that occur in human cells, cancer cells, and how these changes can help develop the next generation of effective targeted therapies. So basically, his lab focuses on how to improve the treatment of cancer patients. Dr. Dutt serves on the editorial board of PLOS ONE, BMC Genomics, Swiss Medical Weekly, Frontiers of Oncology, and in the scientific advisory and selection committee committees of various government and private organizations. He's also a faculty member in the medical genetics section of F1000 Prime. He's a recipient of numerous awards, including the Swiss National Science Foundation Fellowship Award, the Julius Claus Foundation Award from the University of Zurich, the Ramalingaswamy Fellowship from the Department of Biotechnology, the Wellcome Trust DBT India Alliance Intermediate Fellowship, YIM Boston Young Scientist Award, Outstanding Alumni Award from JMI, and one of the most prestigious awards in India is Shanti Swarup Bhatnagar Award in the field of medical sciences in 2017. So Dr. Dutt works on the interface of how infections and somatic changes talk to each other in the pathogenesis of cancer. And so without any further ado, I would like to invite Dr. Amit Dutt to deliver his talk. Welcome, Amit.
Yeah, thanks for that elaborate introduction. So I would start my screen sharing. Can you see my slide?
Yes, thank you.
Perfect. So thanks to the organizers for having me and to tell for the introduction, the kind introduction. So yes, I come from Tata Memorial Center, one of the basic arm of Tata Memorial Center. And today what I'm going to be talking about is about the application of informatics in the clinics, as what the session is all about, with regards to two aspects. So I will be talking more with aspects to the somatic mutations that underlies the cancer genome, and how we can make it, how the work that is being performed in the laboratory has contributed to its translation into the clinics. And then another interesting aspect that is something of a long time interest of the laboratory is also, but something comparatively, relatively much less explored as of yet, is the association of pathogens that is associated with the human cancer genome. So I start with setting up the tumor over here, that why exactly this cancer research is something which is so much kind of like wanted at this point of time, in state of want, as compared to several other diseases. This slide is an old slide, but it has been intentionally kept to be like old. If the slide is basically for the window period between 1950 to 2001, as what you can see, and what you see over here is the drop in the mortality over this window period of 50 years. And if you would compare across different diseases, whether it is cardiovascular diseases, cerebral vascular, or infectious diseases, there is a significant drop. However, if you would, in this time period, window period, if you would see into the cancer, there hasn't been any significant change. And this why is it so? This is primarily this aspect. This part has happened because of the more detailed understanding of the underlying genome, whether it is cell wall synthesis or cholesterol biology. But similarly, in the case of like cancer, we're still scratching at the surface. Not so as much as what the recent data. This is one of the first times in 2019 that there was a drop in the mortality in cancer as well as compared to the other diseases, as what I just showed in a slide earlier, where there was a drop of like about 27%. And this kind of like continues. If you would see even for the 2020, 21, it has not increased. The drop kind of like continues. However, something which is very striking is that this drop in the mortality in cancer is somehow restricted to the countries with a higher income group, and the countries with a middle or lower income group, wherein the India falls, India and China, this you don't get to see this drop in the mortality. In fact, if you would see the cancer burden, the way it is increasing over the time, what is being projected over the times to come, that majority of the increase in the cancer burden across the globe will be contributed by the countries with a middle or low income group, such as India and China. And while in the countries with a higher income group, as what we already saw, there is already a trend of like a decline that is kind of like setting up. So that was something that really places the cancer research and someone kind of like yesterday was talking about that the biology is so important, so relevant that it cannot be just left to the biologist. So I'm a biologist, but then I definitely see that there is a great scope, or the street could only perhaps could like advance if there could be a multidisciplinary approaches, and people, especially with the informatics background, could be could help and try and understand the complexity that is in the lies. So I would set the whole premise over here with some clinical example, and we will talk about some discoveries with approaches to the cancer genomics, and then I would move on to the clinical aspect to it. So here is a real case scenario. Here is a patient who is a 9-year-old girl who presents herself to the clinic with some fever and bruising, and a bone marrow test was performed that revealed a leukemia, kind of a blood cancer. Standard treatment was given to the patient, and the patient enters into remission. Even after 12 years, the patient is live and in a complete and full state of remission. This is usually you tend to get to see in the pediatric cancer. In the same center, under the same hospital settings, the same clinicians treating another patient with a similar clinical manifestations presents himself. Here, this time it's a 7-year-old boy with fever and bruising, and the same test reveals leukemia, same treatment given by the same doctors. Unfortunately, the patient does not really kind of like respond to the treatment. So what does this kind of like really tell? This tells that the clinical representation of the disease, perhaps the least that what it tells us, that the clinical representation of the disease is surely not adequate to understand the underlying complexity, and then there is much more than just the clinical classification based on the clinical representation of the patients, and that's what requires a detailed interrogation into its genome, as what we understand today, that cancer is a genetic disease. So what exactly that it means, that cancer being a genetic disease? Here for the students, just have like going back into the basics of the above. So here in a normal cell, a normal tissue in the human organ would always have a defined number of set of cells, and these cells, they divide in a very defined manner to define a very finite and always an invariable number of like cells. And how does this division actually, it's not that they are always, it's not that the liver or the kidney is always kind of like expanding, we know now, that the division of these cells are pretty tightly regulated. They respond to a survival signal that asks it to proliferate. But at the same time, there is a balance, counterbalance to it, to parallel another kind of like a signal, which is a death signal that asks the cells to stop. This is how what we understand from the development, what we can tend to get to see in the state of like cancer, where the genome receives insults and it acquires mutations and different kind of like alterations. We often tend to get to see is that the cells become independent of the signal. Now the cells are not dependent on the signal anymore, and the cells start to proliferate even in the absence of the signal. The other way, what can kind of really happen is that the cells stop to respond to the death signal, right? And what we get to see in cancer more often than not, that both of these kind of like alterations happen to the same cells, that and that's kind of like really bad. And what makes cancer as like a distinct disease from like other different diseases is that it's in the same individual. You're having two different set of genomes. The cells which is harboring 20,000 or 25,000, the way you look at it, the number of genes. In the same individual, you would have cells, because cancer is a post-birth or it's a somatic disease. The same individual is harboring more than 20,000 or less than 20,000 genes in its genome. So coming back to the patient, that what we kind of like we're talking about, the patient one that did not that responded to the treatment, when it was more interrogated, it was found that this patient harbored a mutation, which is a chromosome 12 fused with chromosome 21. And now we know that most of these kind of like patients who harbor these alterations, they tend to survive. There is a 90% survival associated with them. The another mutation that the second patient that what I saw was unfortunately harboring, and another kind of like an alteration, one of the rare ones that usually is seen in the chronic lymphoid leukemia, that is chromosome 9 to 22, and this has again gone on to the textbooks, altering the gene BCR-ABL. ABL is a tyrosine kinase, and these patients do not respond to the chemotherapy. However, what we now kind of like know from all the work that has been done, you may have read about in your textbooks about specific inhibitors targeted against this BCR-ABL, primarily against the ABL tyrosine kinase, that is the Gleevec. And the success story of Gleevec has been that it has been introduced and approved in 2001. There is close to 100% response. So all of these patients now respond to the a single oral tyrosine kinase inhibitor that is being given to them. So what it does tell us that such kind of understanding these alterations and stratifying the patients based on the underlying alterations, the treatments of these could be given. But then the question that kind of like really arises, that what really makes these individual these cancer cells in particular respond to such kind of a treatment. So here I'm introducing you a concept which is called as an oncogenic addiction. Given that all of these kinases are present in a normal cell and is required for development, in a normal cell, if you would inhibit the activity of one of these enzymes, the cells, given that the different kind of like kinases are present there, more often than not, you tend to get to see a redundancy, a functional redundancy, such that even if the kinase activity is impeded, is inhibited, you would see other cells survive. However, in the case of cancer, what we tend to get to see is that this, most of these activating signaling are so strongly activated to an extent that the cells now are solely dependent on it. And that's what comes the concept of oncogenic addiction, such that now that if you would inhibit this kinase, there is not much of redundancy left in the cell, and the cell tend to kind of like succumb to the or respond to the or is sensitive to the inhibitor. So these kinases, they become essential for their survival. So and then also becomes a pharmacological vulnerability for the cell. So the whole approach has been the way things since the Gleevec has been introduced since 2001. The field has been moving on and kind of understanding more and more of these kind of pharmacological variability or vulnerability that we would be able to identify by sequencing. So such that if there now that we are in a post-genomic era, here is the normal genome sequence, and if we would sequence the tumor, and if you're able to identify these kind of alterations, whether it's a fusion event, whether it is a mutation, whether it's a copy number alteration, these alterations would give some kind of pharmacological vulnerability by and design a treatment against itself. So that's how the kind of like the field set in, the field kind of like started. This was the time when I was kind of joining my postdoctoral studies, and at that point of time, people were involved in the laboratory of sequencing one of the first in a targeted manner of kinases in lung cancer, and they found that patients who harbor EGFR mutation, a tyrosine kinase, in lung cancer, they respond to tyrosine kinase inhibitor, Gefitinib, and this response was seen to be dramatic. What another interesting aspect that came out from the study is that the EGFR mutation frequency, it varies across ethnicity. So that's the variability in the genome. So we still do not know what really accounts for it, but we know that EGFR mutation in the Caucasian population is around 10%, and so is the response to the EGFR inhibitor in the Caucasians, and in the East Asian population, it's about like more than 30 to 50%. We were one of the first ones to establish the EGFR frequency in the Indian population. We know it's somewhere intermediate, 20 to 23%. And so is the response that what we kind of like get to see in the Indian population. So here is the response that you know. Here is a CT scan of lung cancer, and here you see these nodules before treatment, and it does not even take 2 months actually in the clinics. When you give these patients who are harboring EGFR mutation in a matter of a few weeks, you start to see the response. These nodules are gone. This patient harbors an EGFR mutation and was given an EGFR tyrosine kinase. So basically, this is kind of like set the tone for the personalized therapy, or you call it the precision therapy here in this case, wherein you identify these alterations. So with that philosophy, my laboratory at ACTRAC at the Tata Memorial started, and we started investigating several different cancer genomes, because not much have been kind of like reported, and not to the resolution as what we would want to, that would be required to find these alterations. And why specifically you would want to study an Indian specific cancer is for the reason is that they are very significantly different than what you tend to get to see in the Caucasian population. Even in the with regards to the incidence, if you would see, oral cancer happens to be the most common cancer here in India. This is a single site that is being shown over here. If you would combine these and more of like these sites which fall under the head and neck or oral cancer, that happens to be one of the most common cancer in the India. However, that does not really figure as to be like one of the most common cancer in the West. Now, something which is tobacco-associated oral cancer, which is so common among Indian men, is not something that the West is going to emphasize upon. Similarly, among women, cervical cancer is now ranked at number 2. but till a few years back, this was among one of the most frequent, most abundant, predominant cancer among women in India. Now, cervical cancer does not even figure in the top 10 in the Caucasian population. What does that really say? That says that the initiative to understand these kind of like pharmacological vulnerabilities that I just talked about, and if you would want to bring the era of personalized therapy or precision medicine in India, you would need to characterize these genomes, and the initiative has to happen from here in India. And my laboratory has been spearheading and characterizing several of these genomes. What is also important to kind of like emphasize over here is that if you would see from 1990 to 2016, while India incidences has increased 0.5 to 1, the deaths have increased from 0.3 to 0.8. Even Binay yesterday was emphasizing onto these facts with his oral cancer data that even if we tend to get less cancer, 1/3 of the cancer incidence as what you get to see in the Western world, we are more often to die with the disease. So definitely we require to kind of like make broad initiatives in there. So my laboratory has primarily been focusing on several of different these cancers: lung cancer, oral cancer, gallbladder cancer, thyroid cancer, cervical, breast. Today, what I'm going to do is like I'm going to talk about one of the major discovery from the laboratory in lung cancer. I wouldn't talk about the major leads from other cancers, but I would talk about the pathogens associated with across different cancer types among Indian samples. So here I start with lung cancer, wherein one of the first graduate student from my laboratory, he decided to look for mutations in lung cancer beyond EGFR that I just kind of like talked about, EGFR, KRAS, and other known alterations. And what essentially he did was takes two sets of samples. One he calls it as a discovery set, and another one calls as a validation set. This is 125 odd, where he performs directed an NGS approach, and then he validates using mass spec approach across an independent 400 samples. I'm not taking you into the details, but what comes out is one of the first landscape of the resolution that is contemporary to these times from the Indian individuals for the first time. And what we find is that majority of the alterations that what you get to see in the Caucasian population, these genes, you would expect them to occur in the Indian patients as well. But albeit an important observation that what we made, and this now we have completed several hundreds of sequencing performed across different tumor types, and we find something similar trend across, whether it is lung, cervical, breast, oral cancer, specifically the tongue cancer, gallbladder, and other thyroid cancer, that what we have kind of like sequenced in the laboratory. These major genes remain same. They occur at a variable frequency, as what you can kind of like see over here. The frequency of these major alterations occur at a distinct. In fact, what one of the major discoveries that came out from this study was we found an additional mutation in the Indian samples that at that time we were calling as an Indian specific alteration, and that was occurring at a frequency of 5% in another tyrosine kinase, similar to EGFR. It's the FGFR3. And why is it significant? It was significant because 5% of mutation in lung cancer would mean more than the combined burden of, like, say, Hodgkin and non-Hodgkin lymphoma put together. So this 5% in lung cancer is a considerable disease in its own. And what we found is that FGFR3 specific mutation, they were driven by FGFR3 specific mutation. And since then, it has been reported now in the Korean population and others as well. The aspect that I'm not going to emphasize today is that when we find these kind of like alterations, laboratory pairs, my lab pairs, these discoveries with functional studies, strong functional group, where we bring it all the way to the animal system, to the mouse, and we validate and show that the mutations which were derived from the patients, when it brought into the mouse, whether it forms the tumors or not, and when they were given inhibitors, whether they respond to it or not. So those were based on the xenograft studies. And since then, there is an interesting study which has already been kind of like published, where they talk about here is an head and neck cancer tumor, and this patient was harboring FGFR2 mutation, another FGFR mutation, and this patient was given Pazopanib, which is an FGFR inhibitor, a PAN inhibitor, which also inhibits FGFR3, and you could see the response in just weeks. The tumor, the state of like remission. So this kind of like makes it very exciting that the discoveries that what based on the approaches that what we took, if that could be brought onto the clinics. So that is the clinical trial is currently underway that we are going to now we have kind of like validated that or validating that for in orthotopic mouse model. So earlier what I showed you was a xenograft where you put it in under the skin, and now we have gone more sophisticated wherein we directly form the tumors by injecting it straight onto the lung. And what we are observing are the phenotypes, and these cells, they have luminescence associated with them, the luciferase, and what you're seeing is that it's only the lung which is kind of like getting affected. And I'm not looking into the details, but I really want you to know that these white marks, the tumors that what we tend to get to see, the tumors that what we are seeing in the mice is something which is comparable to the tumors that what we even by the phenotype, the clinical phenotype is what we get to see in the in humans. In fact, not only the tumor, what we are also seeing is that there is a secretion of pleural effusion. When we take the pleural effusion, these what we show over here, and when we look into when we do an imaging on that, what we find, the luminescence in here confirms that the cells, these are the tumor cells which are being secreted. Now, this is something exactly that what you tend to get to see in the patients as well. So this really, and this validates the hypothesis that what we had generated based on the unbiased genomic studies that what we had initiated in the laboratory. So this sets the tone for the clinical trial. But since then, the laboratory has kind of like been doing these approaches in several of different. So what I talked about just now was in the adenocarcinomas. Another subtype is lung squamous. We have presented like one of the first, so already published. We have presented one of the first lung squamous landscape of like alterations and therapeutically relevant for which there is no. There is no therapeutic option, targeted therapeutic option available, but based on our studies, there are some interesting leads that what we find, and similarly for the small cell cancer. So, in the clinical informatics session, I wanted to set that base that how a discovery from a genome could be made clinically relevant. And now, in this part of my talk, what I'm going to talk about, another passion of the lab, another interest, long-term interest of the laboratory, besides the somatic alterations, are the pathogens. That's now there is more and more incidences, more and more. The higher resolution we kind of like get into the genome, we kind of get to see non-human genome associated sequences out there. And now we know that there are about like 20% of them of the malignancy across the globe is caused by pathogens, of which 12 to 15 are already by the viruses. We already have heard a great talk on COVID, but here we are seeing the viruses which kind of integrates and transforms the cell. So we were interested in studying. We were wanting to study these pathogens in the Indian samples, and we wanted to see if we could find something which are associated with the Indian cancer, in the cancer among the Indian patients. So historically, how it is classically being done in a conventional manner is shown over here. You require an a priori knowledge most of the times. People have been performing either it's an antibody assay or a qPCR where you need to know the sequence. However, very recently, people have been performing 16S RNA sequencing non-all as well. And more recently, people have developed computational subtraction tools such as VirusSeq and PatSeq that allow, but these tools are pretty complex. They require third-party tool and they're very, very resource intensive. What we started to do in the laboratory made some GUI-based tools, which are easily downloadable, but at the same time could also be done on our server, and which is a much more simplified concept where we made an abbreviated genome. The first tool that what we made in this was, we call it as an HPV detector. The HPV is a human papilloma virus, which is associated and causally associated with cervical cancer. And there are 140 or different strains. But the detection of it is requires a cumbersome process. So we made a tool, HPV detector, that allows you to use the tool to detect the presence of HPV traces in your any kind of NGS data that you have, whether it is exome, whole genome, transcriptome, or even a targeted sequencing. This can be done by a trainee student in the laboratory. And essentially what it does is that it has a reference where we have all the 143 odd HPV genome along with the human genome. It aligns to it and it runs into mode, a quick mode, where it quickly gives you an answer, a binary answer. And at the same time, it also kind of like gives you what are the integration sites of these HPV that is integrated in the human genome. So this work is already kind of published. If you would be interested, I invite you to visit this. And since then, we have been developing many different variants of it, and we have been making and looking for different pathogens. So we look for the non-salmonella typhi associated with gallbladder cancer by looking at the exomes of gallbladder cancer. More recently, along with Dr. Viti Arora from NCBS, we looked into the Merkel cell polyoma virus and so on. What I'm going to talk about in the next few slides is something more interesting and something more exhaustive that what we are currently working on. And this work is still unpublished as what we are communicating the cancer aspect to it. So here we developed a tool that we call as an infectious pathogen detector, and it has 3 different modules. In the module A, one of the module, what it does is we understand about the variability of NGS traces, that is from the different platform, whether it is a second generation, next generation, or third generation, where there could be short reads and long reads. So what it does is that it takes either one of like these 2 reads, and we have made a reference of 1,060 odd pathogens that is known to be associated with human, and then we perform the alignment based on HiSat here for the short reads, and Minimap2 for the longer reads. And essentially what it does in the module 2 is that it quantitates those reads, and we have another reference file which is for the pathogens from the NCBI, where it kind of like takes care of all the different off-target effects, and at the same time it also makes the variant call. Very recently we published a part of it wherein we also added the SARS-CoV-2 analysis module to it, even though it was not intended for that. But we tried to look, because as Vinod was just kind of like mentioning about the genome sequencing, we had a huge amount of like samples which constituted the pre-COVID era proband and the post-COVID era proband as well, and that allowed us to do the comparison and look for the enrichment of these mutations and the occurrences of these traces. And this something is kind of like recently published. This is for if you would want to know more about it. This is what it is all based on, and this is again published where with regards to the SARS-CoV-2. But what I'm going to talk about today right now is how we are how we are interrogating the different pathogens using across. So what we have done now here is like few of these exomes that has been done in the laboratory and in collaboration with Dr. Murli Basham at CDFT for the colorectal. And so there are several more exomes which has been done in the laboratory. Here is just 192 of those that what we take, and the transcriptomes which has been done in the lab. I mean, the lab has now sequenced and has data. These are all Indian specific cancers, data from more than 300 samples, more than 300 samples across different tumor types. So when we look into these four tumors, that is the cervical, colorectal, gallbladder, and the tongue cancer, what we and we set to look for the pathogens which are associated with it. So first of all, what we do is we normalize it, and we kind of like look for that whether the total microbial burden across different samples are comparable or not, because that normalization is something which is kind of like very important. And I just want to kind of like highlight something over here. For example, in breast cancer, we find that the microbial burden was high. Microbial burden was significantly high, and however, we will see that some kind of like surprise that is to come now. So here is based on the exome data across different these four, and here is at the genus level, we have kind of like merged all the species. What we found something as a surprising and something that what you would expect to find. For example, in colorectal cancer, there is a bacterium, Fusobacterium, which is causal to it and or associated with it, and there are significant reports about it. So in the exome data, we could not find much significant association of those. However, we found some significant association of Fusobacterium in the oral cancer, but this was in the exome data. We can talk more about it, but we went more in deeper into it for the transcriptome data. And when we looked at the transcriptome data, and then we also included, along with the Indian samples, to the TCGA data, what we find, and here is now the Fusobacterium, and as what you would expect to find, we find a significant incidence, this heat map representation, where each column is a sample, and each of these rows are the different pathogens. And what you're seeing over here, and this is again at the genus level where the species have been merged. So what you see over here, something on the expected lines in colorectal cancer, significant incidence of Fusobacterium in these colorectal cancer. We find colorectal Fusobacterium in colorectal cancer. We find Fusobacterium also in cervical cancer, which is also kind of like known. What we find, something surprising and interesting, is the occurrence of Fusobacterium in the oral cancer, in head and neck cancer. Now, there are a lot of like commensal bacteria out there in the oral microbiome. So you would need to dissect it out and to see whether this is something similar to what you tend to get to see in the colorectal or not. What was surprising and what was very interesting, that what we saw was the mutual exclusivity of another pathogen which is known to occur in the in oral cancer. We found that samples which harbor or samples which are associated with HPV primarily do not have Fusobacterium, and those that was having Fusobacterium excluded the HPV infection. And our detailed investigation kind of like allowed us to understand better the underlying biology that is associated with it. So here is just validations of those. I mean, as what you would definitely any of these genomic kind of like study based on the real-time PCR. And again, we kind of like see that in an oral cancer, whether it is now even in the exome. As what I said earlier, in the earlier exome data, we were not able to capture for the reason, I mean, exome genome is still an abbreviated. We're just sequencing 1 to 2% of the whole genome, right? But in the transcriptome, we are sequencing all the transcripts. So the likelihood of finding pathogens which are associated and which is also expressed is higher in the transcriptome. We found a significant association of presence of Fusobacterium in colorectal cancer, that what we would expect to find. It was also there in cervical, and then we could validate what we see in the exome, what we could validate also in the transcriptome of the oral cancer. This is a cartoon that kind of like really depicts of the major pathogens which are known to occur across. And here is what I was talking about, is that the samples which primarily harbor HPV do not harbor Fusobacterium in oral cancer, and which is very significant as well as to what we observe in the laboratory, and then they have distinct biology that underlies, and that's what the laboratory is interested in, and that's what we are kind of like revealing. So even when you go back and when we look at the TCGA samples, we find that, and when we would look at what we did is in our strategies, like we took a group of samples which are Fuso high, and here is a group of like samples where there is no Fuso, and then we went around and comparing these along with the HPVs. And what we find is that when we do an expression analysis, we find that here are the genes which are upregulated in Fuso only. Here are the genes which are upregulated in HPV only, and you see there was not a significant, or there was hardly any overlap in this. In downregulated, we did find something, but which is again kind of like suggesting that there is a distinct downstream mechanism or a consequence which is associated with it. Today, I'm not taking you into the details of it because this is more of an informatics talk, but I'm happy to talk to you and discuss more in detail should you be interested. Not only at the gene transcript level, we also looked at the microRNAs, which are distinctly associated with HPVs and Fusobacterium. And we find some microbacteria, some microRNA, which are very strongly correlated, significantly correlated with the Fusobacterium. And given that we have the samples in the laboratory which are Fuso high and Fuso low, we have also performed the validation and looked at the targets. And what it kind of like really reveals to us, or what we this paper is being communicated, it is an unpublished data. What we understand now is that these Fusobacterium, they are associated with some kind of like an inflammatory response management, and that has a therapeutic relevance. When we do the cyber of kind of like analysis, and when we look at the micro, because we know that these pathogens, they affect their microenvironment, we find that there is a pro-tumorigenic association of these cells, of the immune cells, which is again strongly suggestive of they're involved in the pathogenesis, that we are again able to, whatever genomic analysis that what we're doing, we are again able to kind of like validate these by the real-time PCR. Cutting a long story short, there is much more to it, but I just want to tell you that what would be the take home from what we are finding is that these, we are able to find a sub group of head and neck cancer where the Fusobacterium occurrence, when it occurs, they are associated with poor survival, as what is shown over here. And we know that HPV infection is a prognosis. I mean, Vinay also yesterday emphasized onto it in oropharyngeal cancer, that kind of like already predicts, at least in the Western countries, they use it as a prognostic marker about the presence of HPV, but no one is really kind of talking about the Fusobacterium. And this paper details more about the association of Fusobacterium and its consequence downstream, as what could be the mechanism that it might be kind of, So that's something that is a major part of the work that we are communicating this week. So if I'm left with some time, so I just want to kind of like now bring it back. So the first part was the discovery at the mutation space, that is the FGFR3, and similarly that we have done across several different cancers. I've only talked about lung cancer. The second aspect that I talked about the landscape of pathogens. And now what we are tending to get to see is that how these informatically, how we can kind of help the clinicians or bring these findings translate into the clinics. What we know as of today is that despite all the discoveries that are happening, the extent of patients who are really benefited with the precision medicine or with these therapy or mutation-directed therapies, it's not more than it's a very minuscule number. It's only 4.9, about like 5%. It looks like in the clinics are only benefited with these information with regards to the mutation. Majority of the patients, there are several studies now being done that does not really kind of like benefit, and that's not because this is saying that the biology there is an issue, but there is a big gap. There is a huge gap between what is being done in the basic sciences and what is really kind of like getting translated. So that's where to fill up that gap. So we have come up with a newer tool, Clinome, and that integrates the pathogens and the mutations and presents to the clinicians in a way that they can understand it better. So what you would see is a difference in the perspective. So here is, for example, low-grade glioma. If you would ask the clinician, and there is now no FDA-approved treatment. There are several of those cancer types for which there is no treatment at all. So if you would see, there is no treatment as shown by the red circle over here. However, if you would talk to a scientist, the scientist would say that several of these patients they harbor, and that can affect to more than 80% of these patients which are harboring therapeutically relevant alterations. So in principle, the treatment, if approved, could affect 80% of these, could benefit 80% of these patients. However, while we are seeing only 5% that we get to see the benefit in the clinic. Similarly, in thyroid cancer, the same thing. In breast cancer, we do see some targeted therapy that has gone into the clinics, whether it is Herceptin, trastuzumab, or whether it is a PIK3CA inhibitor. But then if you would talk to the scientists, they would ask, and with all the characterization that has been done, there is about like close to 60% of the patients could benefit by these kind of like alterations. So there is a huge the gap between the clinicians and the scientists, and how can that information be made useful into the clinics with the treatment which is approved for cancer A, and it could be offered in an off-label setting into the cancer B. And now that there is more and more receptivity in the clinics for performing targeted therapy, where people are also performing exome sequencing, and they are also performing an extended number of genes to be sequenced in the clinic. But then how to really interpret them is something that is kind of like a huge challenge in the clinics, and that's one of the reasons why there has been kind of like a huge gap. So a tool made by Pratik Shadrani, now he's a faculty. He was a graduate student and a postdoc in the lab. Now he's a faculty at Tata Memorial. Developed a tool which we call as a Clinome. The clinicians, all they need to do, they can take any kind of like an NGS data, whether it is again a whole exome or whether it is a targeted therapy. And when they receive the FASTQ files, they don't need to do anything. All that they need to do is that click OK and tell where the files are. This is still again an unpublished, but something which is already being used in the clinics at the Tata Memorial, where the clinicians are able to analyze it with the raw files that they get from any of the service providers, and if there is and also doing it like in-house. So essentially, what you get from this is the kind of like report that I just showed you. That just kind of like shown up. So essentially what it does is it does a computational subtraction similar to what we were kind of like doing for the pathogens, but this time what it does is that we have made a reference base, a comprehensive reference base of all those alterations where there is already an FDA approval for the treatment, where there are clinical trials which is kind of like going on, and for those alterations where there is a strong evidences that they are likely to benefit the patient. And when they run when they run this tool, the kind of like an output that what they get is something I'm going to show you in the next slide. This tool for the audience primarily here, this also kind of takes advantage of AI, primarily at two different solutions, AI-based solution. It kind of like incorporates. One is that it takes the natural language processing with regards to there's a lot of variability with regards to the cancer type nomenclature. Whether it is called as a renal papillary cell carcinoma or papillary, I mean, different definitions. So what it does is that the natural language processing, it takes any of these and for defining the cancer type. And similarly, what the other solution that it takes is from the tumor type tree, hierarchical tumor type tree that it generates, and which is primarily again for identifying the most appropriate therapeutic target or therapeutic treatment, which is kind of like available, that it kind of like gives as a printout and as an output. So based on these two AI solutions, what we tend to get to see the kind of I'll take you to one of the reports that it generates at Tata Memorial. This is one of the this is a hypothetical report, but what it does to the clinicians only click OK as a button, and this is the report that it automatically generates. Here is a patient which is harboring an endometrial cancer. Again, this is a hypothetical report, and what it does is that, assuming that it finds an EGFR mutation, as what I said earlier, if you would remember, EGFR is a gene for which there is a treatment in lung cancer and not in endometrial cancer. So what this tool, it gives you other different options as well. So it tells you that US FDA approved treatment is blank. There is nothing in here. It automatically kind of like populates these sections, but it tells you that there is an off-label possibility of using this compound, that is a gefitinib, which is actually approved in lung cancer. It's a very pragmatic tool. It does not ask the clinicians to do what, it only states the fact, and then it goes on to kind of like talks about the other different aspects of details of it, about the allele frequency, and we keep educating them, and we keep kind of like sitting on the tumor board and discussing about the coverage, what needs to be looked at, and it has all those kind of like detailed information. So here are just the different cases, different incidences. So here in the lung cancer, this patient harbors 2 different mutations. Now, having 2 different mutations, this is a resistant mutation in the same individual. So what this gives out as a take home is another compound, which is a 2nd generation treatment, osimertinib, but then it also tells the clinician that it just states the in a pragmatic manner, that the 1st generation treatment, even though it's against the EGFR mutation, will not be effective. So you need to preclude that treatment. Similarly, here in this case, there is an FGFR3 mutation. Now, there is no treatment for FGFR3, but as what I said, there is a huge gap. So what do clinicians do? So it links out to the clinical trial, and then it also gives all the relevant references, and then that could be discussed, that could be brought up into the tumor board, and where the scientists and all, they could kind of like basic scientists and clinicians sit together, as what I do at the Tata Memorial Tumor Board or at the Apollo Hospital Tumor Board, and we deliberate on to like these. So here is a case of.
I think they're slightly over time, if possible.
Okay, I'm sorry. I mean, in just a minute, I'm winding it up. So thanks, Joe. So here in oral cancer, there is, so this also integrates the information for mutations as well as pathogens. So here there is an HPV, and similarly, it kind of like talks about. So I would cut short with this. So we have already validated across 40,000, several more of these samples. This is the last slide. What I really want to tell you is that this tool, which would be downloadable soon and publicly available, it has been transferred to the companies, to an industry which has shown an interest to an MOU, where here is of the 40,000 samples that what I showed in the earlier slide. Here is the extent of samples which would be for which there is an FDA approved treatment, and then this information that it gives to the basic scientists who would be interested in those alterations for which there is no treatment as yet, but then from the patients, it identifies these alterations and that can be taken up accordingly. So with that, I bring an end to it. Sorry for extending my time. We talked about the application of informatics in high throughput data analysis and making discoveries which are clinically relevant. I only talked about lung cancer, and then we talked about the pathogen landscape. That is an upcoming story that we are communicating this week. And we discovered Fusobacterium association with that. So, and then the tool Clinome, where we integrate pathogens and mutations and present it up in the clinics. This is, I mean, a big group, and of course, most of the work that has been kind of like done are the people who are kind of like shown over here. So our laboratory is very well funded thanks to the Dutt Memorial Center, Department of Biotechnology, Welcome to DBT in the Alliance, DBT VNCI, and the Terry Fox Foundation. So yeah, with this, I bring an end. Sorry again for extending my talk, and should there be some time and questions?
Thanks, Amit. So we'll take one burning question. I think there's a question from the audience. Professor Dhruv Kumar